Assalamualaikum and hi. We're from Group 4 for Introduction of Genetics. I am ROSESYAQIRAH BAIDURI bint RAZAK and my metric number is F20B0586. For this assignment, we are assigned to talk about one subtopic for each chapter, and we have chosen to discuss about the variation in chromosome structure. Firstly, what is a chromosome? A chromosome is a strand of DNA that is encoded with genes. In most cells, humans have 22 pairs of these chromosomes, plus the 2 sex chromosomes, which is XX in females and XY in males, for a total of 46. The word chromosome was originally coined in German from the Greek words of chroma, meaning color, and soma, meaning body. Next, there are two primary ways in which the structure of chromosomes can be altered. First is the total amount of genetic information in the chromosome can change by if the number decrease, it is from deficiencies or deletions. If it is decreased, then it is from duplications and insertions. Secondly, the genetic material may remain the same, but it is rearranged, which is by inversions or translocations. There are 4 common types of chromosomal mutations involving changes in chromosome structure. Firstly, there is deletion, and then duplication, third, inversion, and lastly, translocation. Next, we will talk about deletion. What is deletion? Deletion is a chromosomal mutation in which a part of a chromosome is missing or deleted. Since a segment of chromosome is missing, so deletion mutations cannot be reverted to the wild type. Deletions start with chromosomal breaks induced by heat or radiation, viruses, chemicals, transposable elements, and errors in recombination. Deletions do not revert because the DNA is gone or degraded. As you can see from the figure, the segment D is deleted from the chromosome, and soon it will form a chromosome without segment D. When parts of chromosome are missing, a number of syndromes can occur. These syndromes are called chromosomal deletion syndromes. They tend to cause birth defects and limited intellectual development and physical development. In some cases, defects can be severe and affected children die during infancy or childhood.
Duplications. A duplication is a chromosomal mutation that results when a segment of DNA breaks off and is attached onto the homologous chromosome. There are different forms of chromosomal duplication or different tandem configuration. 1. Tandem. 2. Reverse tandem. 3. Terminal tandem. Tandem duplication are adjacent to each other. When the order of genes in the duplicated segment is the opposite of the order of the original, the mutation is reverse tandem duplication. If the duplicated segments are arranged in the tandem at the end of the chromosome, the mutation is then called terminal tandem duplication. Duplication can cause phenotypic variation that might at first appear to be caused by a simple gene mutation. The bar eye phenotype in Drosophila is a classic example. Instead of the normal oval eye shape, bar eye flies have narrow, slit-like eyes. Inversion. It is a chromosomal mutation that results when a segment of a chromosome is excised and reintegrated at an orientation 180 degrees from the original orientation. There are 2 types of inversion. 1. Paracentric inversion. 2. Pericentric inversion. Typically, genetic material is not lost when an inversion takes place. If the centromer is not part of the rearranged chromosome segment, it is a paracentric inversion. If the centromer is part of the inverted segment, it is described as pericentric inversion.
So for the translocation of chromosome meaning is translocation is a type of chromosomal abnormality in which a chromosome breaks and a portion of it reattaches to a different chromosome. Chromosomal translocation can be detected by analyzing karyotype of the affected cells. So what causes translocation of chromosome? However, the mechanism which causes this translocation remain poorly understood. Illegitimate recombination, class switch recombination, homologous recombination, non-homologous end joining, and genome fragile sites all have potential roles in the production of non-random chromosomal translocation. So how does translocation happen? Translocation occurs when chromosomes become broken during meiosis and the resulting fragment become joined to another chromosome. Provided that there is no loss of genetic material, the translocation is balanced, no loss or gain of genetic material, and usually results in normal development. So, translocation. A segment of one chromosome becomes attached to a non-homologous chromosome. Simple translocation is a one-way transfer. Reciprocal translocation is two-way transfer. For the A, non-reciprocal intrachromosomal translocation. Non-reciprocal translocation is a movement of a chromosome segment to non-homologous chromosome or region of same chromosome without reciprocal change. Intrachromosomal translocation involves the movement of a chromosomal segment from one location in the chromosome to another. For B, non-reciprocal interchromosomal translocation. Interchromosomal translocation involves the movement of a chromosomal segment between chromosomes. For the C, reciprocal interchromosomal translocation. It's same with B, but in C, it has reciprocal translocation. Reciprocal translocation occurs when chromosomal segments are exchanged between two non-homologous chromosomes, and it the most typical type of translocation. So for the conclusion is variation of chromosomal number involving a set of chromosomes polyploid or an individual chromosome aneuploid can occur. Aneuploid results in syndrome with distinct characteristics.